Hello everyone. Here we are today with our Microtome lab, a lab where we're going to expose you to a very important piece of scientific equipment that we use to prepare our microscopy slides. Because, as you will have experienced in working with a microscope, thinness is the name of the game. If you want to see anything visualized under the microscope, you have to be able to see through the layers of the material. And I don't know about you, but I have a very hard time cutting things that are very tough, very thin. I'm more likely to cut off my fingertips than to cut off a usable section of a stem, particularly if it's of a woody plant. So to help in those endeavors and to work with the slides that we're working with in this lab, obviously we don't need a microtome for blood. Blood can be smeared. But for any of our materials like the tilia stem, as you see in this lab, or any plant roots or shoots, you need a microtome. And this is the basic piece of equipment that you'd expect to find in any lab. In fact, or any lab that does microscopy. In fact, my wife, doing her postdoc in Germany at the Max Planck Institute at the University of Tübingen, had this exact piece of equipment there. So this is lab grade, research grade material that allows you to train here at West Shore and then take your skill set to wherever you go on to in the future. We're going to talk a little bit about what all the knobs and buttons do. I'll have Becca give that little bit of a demo. As a general theory here, you're going to control the size of the sections that you're cutting, catch each of them using a paintbrush as they come down. You can do serial sections, so that way you can get a slice throughout a component. Obviously, you're not cutting something as giant as a human finger, but you can get serial sections through it. And then you're going to lay them out on a slide. And as this slide that's dried out from before shows, you can get things that are thinner than paper, getting down to individual cell thickness. So let's turn it over to Becca to talk about the bells and whistles.
As Paul was telling you guys earlier, in order to use the microtome to cut very tiny sections of something, usually it has to be embedded into a plastic. Now, we can't do that today because we have some of the supplies to do this, but we do not have all of them. In order to actually do this process with plant tissues, we would need to remove all of the water from it and make sure that it's embedded in a plastic, and then we would cut very, very thin pieces of that.
And how long does that protocol take, Becca?
Protocol takes about a week in order to remove all of the water from any cell.
Yeah, that's pretty crazy. So the long 6-day protocol, we'll wait till we get all the reagents. But for now, walk us through all the bells and whistles of this piece of equipment. What are we controlling, what's moving, and what do all the knobs do?
All right, so we'll go from one side to the other. So we start over here. This big wheel here and the handle on it, see it's got a lock. All that this wheel does is it controls the mount. So every time that it spins, the mount is going to move, and every rotation is one more cut. And the lock on it helps you to make sure that it's not just continuously moving and in motion. It's just a safety precaution more than anything.
Perfect.
Along with that, you can see this little guy down here. It's just another one of the locks. So the primary locking position for the wheel is upright. But if you want to lock it a little lower in order to make sure you have everything aligned, you can use this to lock it in different positions.
You want to demo that for us to show the different lock position, Becca?
So when it's either side, one way or the other, that means it's unlocked. Push this in, it unlocks the wheel. Say I want it here. I'll move this up, and the wheel is now locked.
Perfect. Let's go ahead and return it back to its standard upright. You should always leave the machine as you found it, so thank you, Becca.
Mm-hmm. Now coming over, we see our next little twist here. So this one, again, we always want to make sure these are locked. Whenever we're using it, all of these things need to be locked in place. This one, very importantly, controls our blade here. So if it is unlocked, move this here, Paul got a little tight, it will slide our knife back and forth, so we can move it either forward or backward, depending on how large our sample size is that we put into our mount. We're going to keep it out a little ways for now. Next, starting ground up, we have our tray. All this tray does is it catches all of our scraps, because because otherwise we would make a really big mess.
True story.
And all this is it is a magnet that just pulls right out of there. So you can empty it when you're done.
Yep. Whenever you use it, please do make sure to empty it.
Yes, we want to leave things clean. Makes them last longer. All right, moving up, we have our blade. As I showed you, it already moved it back and forth. We have it locked where we want it, but we can adjust the angles on this. So actually, we'll start with this. This little knob here controls where the blade goes in. So the blade is actually going to be right on the top. You can see there's a little bit of a gap there. That's where the blade is going to go. And that just keeps it locked in place for us. Along with the blade, we've got this little guy. This is our safety guard. This is very, very important. Whenever you're not cutting or actively using the spinning wheel to cut something, the guard needs to be up if there's a blade on it, because the blades are incredibly sharp and you do not want to cut yourself on it.
Totally agreed. It cannot be stressed enough that the blade guard is your defense from any accidents. So if you are not actively touching the wheel, the blade guard needs to be up.
Mm-hmm. Along with that, you can see this little arrow and button combo on here. So what this does is if there was a blade in here right now and I had this loosened, has to make sure that it's loosened, you could tap this and it would push the blade out over to this side for us. So you could grab it without having to contact the sharp part of the blade.
Sounds like a safety precaution to prevent you from cutting yourself.
Yes, it is. This lever here adjusts the angle of our blade. So if I loosen it here, you can see that we can adjust. It's a little sticky, but it will adjust the angle of what we have going on here.
Yeah, let's go ahead and not futz with it now. Let's keep it as to exactly where we want it with the two lines flush. We moved it a little bit. Let's see, so there we go. Shifted it down a little bit more and we're back. So let's put that back in lock. You can change the angles on different things if necessary, but for now, we'll just keep it as is. And let's talk about the knobs on our mount.
Okay, so the knobs up here on the mount, we'll start here. This one, this is a locked position, up, as most of them are. When it's up, that means the mount is set in place. Everything is set, we're not gonna mess with it anymore. When it's down, however, we can change our other knobs up here. So this one is just a rotation. Right now it is set lined up, it is perfectly even and level because we can see the red knob right here. That tells us that everything is flush. This is the original position of everything. When I start moving this, this is our vertical tilt. So if we have a section that we ended up cutting a little weird, or if we want to cut it differently, we can rotate it like this, and it rotates it up and down.
So you notice the head piece, this actually tilting down.
Mm-hmm.
Go ahead and crank it a little bit. There we go.
And when I want to bring it back, make sure everything's flush, I'm just going to watch for my little red knob to appear again. There it is.
And there it is.
And back in place. Now, along with that one, we have this one. So this is our horizontal tilt. And it does same idea, except horizontally. So you can see it's tipping right. It'll also go left. And it also has another one of those little red knobs on it, as you can see when you're back in the original position.
Perfect.
There it is.
All right, let's go ahead and lock that back up. Now, how about this dial here, Becca? What's that do?
This dial adjusts how big of a cut you want to make. Right now, it is set to 60 microns, and this can go way, way down, all the way down into very, very, very small numbers, obviously.
So down to a single micron resolution on this, which is quite impressive. For the purpose of these giant cuts today with our carrots, we're gonna make nice, big, heavy cuts, because without the help of the plastic embedding and making it a tough specimen, you can't get that good, small resolution. Last two doohickeys.
The last two. This one is, it's another one of the quick motions for our mount. So if we have it set at very, very small intervals for the microns, the top one will be 10, so a jump forward of 10 microns, and the second, the two dots, is a jump of 30 microns.
So very small in the grand scheme of us as humans, but very big in the grand scheme of microscopy cutting. Mm-hmm. And last but not least, the dial on the left side of the machine.
The dial on the left side controls how far out the mount is from the blade. So it's a very, very quick motion of forward and backward, and it's not controlled in microns at all. It's just so that you can get everything lined up.
Cool. So now that we've gone over all the buttons, let's go ahead and get our carrot sample placed in. So ultimately what we're going to be doing is taking our carrot and putting it into here and getting all of the knives placed into the holder and cut our first section on this microtome. See y'all in a second.
We made a mistake that we now realize. So the angle on this is more of a set-in feature that isn't changed as often. So that is going to be changed over here. There's a little Allen wrench kind of thing that changes that. So what this knob actually does, that we were trying to fiddle with, it controls where your blade is placed so that you can use more than just one spot on the blade.
Yep. Excellent. Now we're going to go ahead and set up that carrot for y'all. All right, so we've locked the carrot in place and adjusted the mount so that way it's close. Let's go ahead and take a vertical view on this thing moving. I think we're directly above it. Becca's going to be taking all safety precautions and doing the actual cutting, so take it away. Still not quite close enough.
Starting to.
Shaving the edge. Oh, I heard something there. And how do we clean those pieces off when we don't want them there anymore, Becca?
We're gonna need either some tweezers or a paintbrush. And we can just get rid of those.
Perfect.
It is cutting it, it's just since it's more of a squishy sample, it's not cutting off nice pieces yet.
Yeah, we'll get it to a point that we cut a nice thick section for y'all. Give us a second. All right, so after a fair bit of effort, we've got our two different sections. So this monster piece was actually cut without using the 60 micron dial. That is way thicker than 60 microns, but it was the first piece to create a flat edge. Still useful, but too thick to visualize because light will not be able to penetrate through that, right? If I look at it from the side, light's not getting through it very well. This flimsy, almost looks like dirt speck next to it, is actually one contiguous piece of tissue. Has a couple of holes because some of these cells are too big, but that will spread out nicely on a microscope slide. And in order to prove it to you, we'll do it now. Again, off camera because it's a little bit difficult to record, but let's go ahead and take a look at what this looks like under the microscope. So, just to prove it to y'all that this is actually the section that you just saw, this is what we pulled off of the side of the microtome. It's incredibly thin. Becca prepared an amazing slide with a pencil drop method, creating no air bubbles, so please make sure that you have that practice for the exam. We're now gonna go set it up under the microscope. You wanna take care of that, Becca, while I hold the camera?
Yeah.
All right. We're gonna go ahead and get it focused for y'all and publish the pictures. All right, so here is what the tissue looks like. You can see that there are some orange chromoplasts from the carrot giving its orange color, but it's very difficult to see any cell features. This, of course, leads to the next component of today's lab. After you understand how the microtome works, you have to understand how stains work. So please do the reading in the lab about the stains, and let's go ahead and take a look at some images. In the meantime, Beck and I will prepare this sample now that you see it, or really don't see much of it underneath general light microscopy. We're gonna stain it, so that way hopefully we can see some cell walls in this. See y'all in a bit for our discussion of other materials prepared via microtome with beautiful stains.
Notice that human blood is not shown under white light because it just looks like a bunch of red dots. Here you see the nuclei stained within the frog or fish blood. Here you can see frog and human blood adjacent to one another. Notice the difference in size of the two cells, with human blood being much smaller, in large part because it lacks a giant nucleus. The nucleus is very visible using the stain that's present on these slides. For more information on that, please refer to your lab. Here we see a tilia twig without much definition. This is about as good as it gets without microscopy equipment and stains. Here we see a 1-year-old tilia stem with a large amount of vascular structure in the middle, and then the beginnings of the first ring of vascular tissue on the outside. For our 2-year-old stem, we see 2 rings. Pith still remains, bark growing thicker. For a 3-year-old tilia stem, we see 1, 2, 3 rings. Pith still remains, bark continues to expand. With a different laser frequency, we see the fluorescence of different cell types based on their chemistry. Here we have a branch of tilia, but interestingly, I wanted to show how, because of the fluorescence generated by this, we actually see an image projected onto the lamp at the base of the microscope. We thought that was pretty cool. Here we have tissue that was not listed in the lab, a sunflower stem. You'll notice the vast difference in lignification, or the thickness of the cell walls. This is herbaceous plant material. The cell walls are not as thick, despite the fact that they are labeled with a similar stain. Compare the amount of fluorescence from these cell walls to the fluorescence of the tilia stem, the tree stem. We also have a zoom close-up of the vascular tissue, the heavily lignified water-conducting tissues, and their support structure, the sclerenchyma. Here we have a slide that Becca prepared without any of the fixatives, so quite impressive. Took a lot of patience to be able to see this Cercis canadensis stem. The labels are not yet in, so our fluorescent dyes are not yet in, but you can still see the difference in the pith in the middle, the vascular tissue. This might be 1 to 2 years old. We have to see with a stain to be able to differentiate those components, but the bark is on the outside there. It's supposed to help inform your answer on the truffula questions regarding self and cross-pollination. Here, I used a stain called aniline blue to identify pollen tube growth. On the left, you see self-pollen growing on the California golden poppy, and on the right, you see cross-pollen. Do you notice a difference in how far these different types of pollen are able to grow, and whether they would potentially be able to fertilize the egg? Consider that when studying or discussing your truffula tree.